Hello, my name is Frank Böckler and I'm the corresponding author of Synthesis of Amino Acids bearing Halodifluoromethyl Moieties and their application to p53-derived peptides binding to Mdm2 and Mdm4. In the last 15 years, we've investigated principles and applications of halogen bonds in medicinal chemistry, chemical biology and drug discovery. The halogen bonding paradigm is based on the anisotropic electron distribution around the halogen atom, leading to positive electrostatic potential in elongation of the CX bond, which has been termed the sigma hole. Nucleophiles in the binding sites of drug targets can form attractive directional interactions with the sigma hole. The negative electrostatic potential is predominantly found as a belt around the halogen atom and can form attractive interactions with electrophiles such as hydrogen bond donors in the binding site. Here you can see the electrostatic potential of iodobenzene in a 3D visualization. Please take notice of the white surface separating positive ESPs in red and orange from negative ESPs in blue and cyan. It highlights the extent of the sigma hole and how easily it can be accessed by a nucleophile. When introducing the halogen into a heteroaromatic scaffold, the sigma hole can be vastly tuned, increasing its strength and expanse while reducing the negative ESP at the belt of the halogen. This typically facilitates that a stronger halogen bond is formed. Using CF2X moieties as halogen bond donors in this manuscript, we release halogen bonding from this two-dimensional confinement and enable the formation of strong out-of-plane interactions. Strong negative ESPs are found at the belt around the iodine atom, as well as enclosing the oxygen and the fluorine atoms. In the study, we've designed various CF2X-bearing FMOC-protected L-amino acids. The first set is based on the phenylalanine or tyrosine scaffold, introducing a bromo-difluoromethyl group, a bromo-difluoromethoxy group, a bromo-difluoromethyl group, or a bromo-difluoromethyl sulfonol group. The second set is based on lysine, or its shortened analog L2,4-deaminobutyric acid. Their terminal epsilon or gamma amino function was converted into the acetamide, the trifluoroacetamide, the difluoroacetamide, the chlorodifluoroacetamide, and the bromodifluoroacetamide. Based on our previous experience with the helical peptide of the tumor suppressor p53 binding to its key regulators Mdm2 and Mdmx, we used the system as a case study for our newly obtained amino acids. Based on calculations, modified p53 peptides were designed and obtained through solid-phase peptide synthesis. Testing of the modified p53 peptides was conducted by a competitive fluorescence polarization assay. If the fluorescence level reported peptide is hardly displaced from binding to Mdmx, the slow tumbling of the complex will lead to a polarized emission over the concentration range of the test peptide. However, if the test peptide binds strongly to Mdmx and displaces the reporter, the fast tumbling of the free reporter peptide leads to a depolarization of the emitted light. The tighter the binding of the test peptide, the lower the needed concentration for depolarization. Here you can see the results obtained for the lysine derivatives, here for the DAP derivatives. The affinity for lysine in position 24 is impaired by the acetylation. The trifluoroacetamide derivative shows some gain of affinity, followed by the difluoroacetamide, which improves the affinity beyond the level of the unmodified lysine. Chlorodifluoroacetamide and bromodifluoroacetamide show even stronger binding, providing first evidence that a halogen bond can be responsible for the effect. The DAP derivatives in comparison lack a similar trend and typically show weaker binding than their longer lysine-based counterparts. This suggests that the length and flexibility of the side chain in TPA6 is crucial for forming an attractive interaction. As it was impossible to obtain crystals of TPA6 with Mdmx, we applied computational methods to characterize the putative interactions. First, we performed an exhaustive conformational search for the modified lysine side chain. Then we implemented all conformations into the peptide and discarded all clashes. p53 peptide and binding site models were derived for human Mdmx from crystal structure 3DAB, and for zebrafish Mdmx from 2Z5T. Geometry optimization for the binding site model was performed applying density functional theory in Turbomol. Some interactions, such as the halogen bond onto glutamine 58, were rarely found. In comparison, the intermolecular halogen bond onto histidine 54 is more frequently found from different starting points in both binding site models. Interestingly, an intermolecular interaction with the charged aspartate in position 21 of the p53 peptide is found very frequently. Almost all obtained halogen bonding geometries show a very good to excellent halogen bond distance and a good to excellent sigma hole angle. From the statistics, it becomes evident that histidine 54 is the unrivaled most plausible halogen bond acceptor in the Mdmx binding site. However, some intermolecular interactions, particularly with the aspartate in position 21, can be major distractors competing for the halogen bond. Thank you for watching this video. If you're interested in our work on halogen bonding, please get in contact.